Good afternoon. This is Dr. Dan Guerra from Authentic Biochemistry Podcast Outdoor Studios, one of the many places I do outdoor lectures. This is a Sunday afternoon, and I believe it's the 28th of August. So I said I was going to do a membrane lecture on video because I thought it might be a good idea for one thing to, so that we can get to see each other, or at least you get to see me, and you know who's talking to you through all those audio lectures, right? Which we normally have as a platform on all the regular places and ways that you might be listening to podcasts. I typically take these video lectures and then reorganize them into YouTube videos and then post them on my YouTube channel. And I'll put that in the show notes of some subsequent podcasts in case people don't know about that. But you should be able to find me pretty easily on Authentic Biochemistry video lectures. Just throwing my name in there and the name of the podcast. You should be able to find it. So let's go ahead and get started on this project. All right, move me out of the way somewhere here. So what I want to do today is kind of a synthesis lecture, but it's also going to give some new information. It's typically how I do it. I'm going to call this sex differences, including natural killer cells, which are part of the innate immune response, and the switch to tissue remodeling. Okay, and I believe it's lecture number 44, a number I like a whole lot, just like the way it sounds. Okay, so let's get going with this. Gonna make me smaller. And that's going to be the ontological synthesis of what we're going to be doing. We have already said, and I'm sure most of you are fully aware, that biological membranes separate self from the external environment. So they are the boundary layer between everything is external to the living system and everything that's internal to it. Hence, they act as a barrier, but also as a semi-permeable conduit for the communication that's necessary, the biochemical communication that's necessary between the external and the internal capacity of any living system, particularly something that is multicellular and highly complex, differentiated and developed like an animal, particularly human, that interaction with the environment is nuanced at multiple biochemical and physiological levels. And that's what we spend most of our time talking about in authentic biochemistry, because I try to focus on biomedicine, and particularly pathobiochemistry and pathophysiology, which means there is some kind of stress response that's occurring external to the human body. And then we talk about the initial phase of how that stress is received, usually via some kind of receptor-mediated exposure, and then all of the subsequent signaling and then alteration of metabolism, particularly through the immune response that follows. Right? So the membranes, besides having that external-internal, very important barrier, but also semi-permeability, designate and isolate domains of biochemical activity. It's one way of looking at it. They do it so as to organize gene expression, signaling, bioenergetics, and of course, the rest of intermediary metabolism. The composition of membranes varies with the function. These are all pretty hard and fast premises. And I think I've already made good arguments for each of them, but these are premises, or we could also call them propositions in the argument about how membranes function. Okay? And I believe these all to be true. So if propositions are true, then your conclusions will also follow and therefore be obtained, so obtained and validated. So if you have a sound and valid argument, that means you're trying to express truths. And I think that's what we're doing here, as much as is possible from my expertise and from what I read in the scientific literature, research scientific literature. Now, protein biochemistry, when we talked about that in some previous lectures you've heard me do, or maybe you've heard it from someone else, unfortunately, we start by talking about amino acid composition, just like we have the composition of membranes, right? Composition meaning you have lipids and you have proteins, and you also have nucleic acids, carbohydrates, right? But in protein biochemistry, we start with amino acid composition, but we don't spend too much time there. What we quickly do, because we quickly developed instrumentation and biochemical separation techniques to discover the protein primary structure. After all, it is a polymer. So it has a sidedness to it. It has a geometry. It has a physics. It has a three-dimensional space and also space-time, right? Because proteins change over time in terms of their folding, reactivity, movement, sequestration, and then ultimately degradation back to amino acids for the lifespan in the cell that they happen to support for that particular primary structure. So that's what we mean by primary structure, the amino acid sequence. And we talk about coming from the amino terminus, which is essentially the beginning of the polypeptide, to the carboxylic acid terminus, which is the end of the polypeptide. Now, we've said this, I've said this many times in biochem lecture for the last 30 years. I know I was really young when I was doing lectures. You can tell. We don't have a primary structure for biological membranes. That is, we don't have a sequence. We know it's there, but we don't have a sequence like we do with polypeptides or with nucleic acids, both DNA and RNA. Or even for that matter, for carbohydrates, complex carbohydrates that are, for example, covalently bound to polypeptide. We have developed ways of breaking apart specific bonds of carbohydrates so that we know what a glycocalyx looks like. We know what a glycosylated polypeptide looks like in terms of how the carbohydrates are structured very specifically. But because lipids pose another element of problem in that they are hydrophobic, so many of the interactions between the membrane and lipids, and you know this if you listen to my lectures, many of those interactions are not covalent bonding, right? Hydrophobic interactions. That is the exclusion of water, allowing for the inclusion of particular lipid species to form membranes spontaneously. Those aren't covalent bonds being formed. And then within those membranes, all the proteins that are captured within the macromolecular structure, which is essentially synthesized from activities of the cytosol, the mitochondrial mitosol, the intermitochondrial space, the endoplasmic reticulum, both at the membrane and internal in the lumen of the ER, and then the Golgi apparatus, both cis and trans organizations of the Golgi apparatus, and then finally even the plasma membrane. There's sometimes more alteration of membrane and protein. Other features that occur there, of course, is gene expression. So the nucleus and nuclear envelope are involved, as well as the peroxisome and the endosomes and the phagolysosomes. So all the subcellular features of the given cell type are involved in the biosynthesis of that primary structure for the membrane. So it's not that it doesn't have a primary structure, it does. It's just that we don't have the techniques to be able to take a look at that. Now, we do have a concept of membrane lipid rafts, and I spent a lot of time giving you lectures on that. And those are basically domains of membrane that are manufactured, to put it simply, between the mitochondria, the ER, and the Golgi and the plasma membrane. And they're trafficked sometimes in and out of endosomal micellar structures but they're trafficked to the plasma membrane and to all the other membranes as well with a cargo of polypeptides that are inserted specifically into subdomains of lipids that it can be something like bilayers, or they could be unilamellar structures, or they can be inverted cones, or they can be regular cones, all kinds of different hex2, hex1 phase structures that we talked about when we were discussing the very beginning of these membrane lectures, right? And within there is embedded specific polypeptides that are essentially co-synthesized with the membrane lipid components of the membrane lipid raft, which is also cargoing, as I said, polypeptides. Not just polypeptides, but right now we'll keep it simple. So we know that lipid rafts, which basically are domains, can also form these caveolar structures, which are important depot domains, which allow for the more vigorous transport of complex macromolecules from external to internal and from internal to external. By that I mean things like small interfering RNA, microRNA, even messenger RNA, and certainly polypeptides and lipids and signaling molecules like neurotransmitters. Okay? All right. So KVLI are a very important thing that we discussed a great deal. I'm not going to do it right now, but that's a key feature of bioactive membranes in living systems, particularly humans. Now, there are specific lipid classes and domains, and they're distinguishable for certain secondary structures, and that's what I was just talking about. So, we have secondary structures of polypeptides, like alpha helix or beta pleated sheet, right? When lipids, and in membranes, we talk about bilayers, which is your classical image in your mind of a bilayer, right? You have two leaflets, you have an outer leaflet and inner leaflet. The interior of the membrane is primarily hydrophobic, where the fatty acyl chains occur. And then on either the outer leaflet edge or the inner leaflet edge, inner leaflet will be the cytosolic exposed surface. Those will where you'll have, if it's a glycerolipid, the so-called polar groups or polar head groups, right? Okay, just to get an idea of that orientation. Inverted cone geometry, something else we talked about. And again, these are all generated by having specific membrane lipid classes and then the subclasses, such as phosphatidylethanolamine versus phosphatidylcholine, or phosphatidylserine versus phosphatidic acid, or diacylglycerol, or sphingomyelin versus ceramide. All of these lipids will generate a three-dimensional structure, which is in constant dynamic event ontology. So it's actually all four dimensions. Time is a major key signature and feature of living systems, right? So the membrane is constantly changing in dynamic. It is not a wall. We have cell walls in plants and in bacteria. We don't have cell walls in animal tissues. We have membranes. We have all those other organisms also have membranes, right? So cell walls really control osmotic pressure and water relations, right? So that's not a thing that you have with organisms that generate their own heat, okay? So the poikilothermic organisms tend to have cell walls. And there's a whole lot of biology and evolutionary biology about that, and evolutionary biochemistry about that I'm not going to talk about here. But I have in the past. If you go back and listen to all the lectures I've done, including the four and a half, almost 5 years, I've been doing it online. But then go back to 30 years, I've been doing it in lecture halls all over the Intermountain West, and even in some places in the Midwest, and in fact in Europe. All right, so enough of all that. There are profound physiological differences of membranes according to cell type. So composition, and of course, whatever the primary structure is. When I begin my primary structure, I mean how lipids are organized associated with one another, because that is very specific. You don't just have a huge amount of glycerolipids and sphingolipids and cholesterol and plasma membrane, and they just sort of organize, and that's what and their composition is almost basically also their secondary structure. That's not the case, because the temporal signature of membrane lipid synthesis is when they're being synthesized and the raft is mobilized. Now, not only that, when you make a polypeptide, you make DNA or RNA, you make that macromolecule, and then it stays as that macromolecule until it gets degraded. There might be some minor changes, like, for example, transfer RNAs, there's more than minor changes, but those are all covalent, right? With a lipid, the membrane itself, which is again a macromolecular structure composed of lipids and proteins, and of course carbohydrates and nucleic acids, all. But the membrane lipid is what creates the structure. And remember, in biochemistry, we talk structure function relationships. So enough said there, right? So it generates, because of lipid heterogeneity, polypeptide asymmetry. So you have some proteins that are more on the surface, some that are almost completely internal, and some proteins that really are exposed more to the cytosolic domains. Or you have proteins that completely ribbon through the membrane. So you have, for example, receptors where they'll bind a ligand extracellularly. The ligand will then alter the protein structure within the membrane, cause it to rotate or move, or to sometimes flip to the other side of the inner leaflet to carry out a signal transduction. Or it's just simply the carboxy terminus that may be in the cytoplasm that carries out the rest of the signaling, but that's altered after ligand binding on the external surface, you see. Now keep in mind those whole membrane domains are constantly changing. That's what's unique about the membrane macromolecular structure as compared to DNA, RNA, and protein. Because there you synthesize it, and again, it's whatever the DNA is doing in terms of replication, recombination, repair. That DNA is going to stay solvent as DNA until it's degraded, right? Same thing with a polypeptide. You make the protein, eventually it's going to be digested in the proteasome, or it's going to be broken down again by exoproteases or endoproteases, et cetera. And then you have RNases and DNases that break up DNA and RNA eventually, right? Down to the monomeric components, nucleotides for nucleic acids, and then amino acids for the peptides and complex proteins. And of course, the carbohydrates do get broken down. But with a lipid, you don't necessarily break down those lipids. You're just constantly trafficking whole domains of lipid with protein to specific subdomains of that membrane on specific cells. That's why you get all this very complex asymmetry. And it's absolutely dependent upon the biosynthesis and the time signature for the movement of the membrane lipid rafts in and out of that membrane. Plus, the normal removal of, for example, highly polyunsaturated fatty acids coming out as glycerolipids or sphingolipids to be partially removed and then utilized either in bioenergetics or used as precursors to resynthesize very long-chain polyunsaturated fatty acids of the omega-3 or omega-6 series for specific biological functions such as neurotransmission. Okay, so it's a very dynamic system, and you constantly are turning it over. And how quickly? Oh, it can turn over in the matter of microseconds. You can have entire sections of the membrane flip from external to internal in that amount of time. But normally it works in milliseconds. It's much more rapid than something like protein synthesis. So protein synthesis might take 5 minutes, 10 minutes from transcription to translation. This can occur in real time. So membranes and membrane lipid components are constantly, penocytically sometimes, testing what's going on in the external environment and the internal environment to be able to modify their structure to deal with the vagaries of space-time, which is happening in the immediate, not in the mediated time signature. You see. Now, on top of all that, we've been talking more recently in these 43 lectures before this one, that age, sex, male or female, and disease obtain specific and unique membrane structure function dialectical events. And when I say dialectical, I mean this or that, and then this or that, or maybe this and that, or the other, right? Either or, either A and B or C, right? And then multiply that exponentially, and you get an idea of what I mean by a dialectical event in the membrane, right? You know what event is. change over time, delta T, right? Of course, that's what I mean. Okay, let's continue on here. Now, we talked about this paper before, but again, this is a synthesis lecture. So I'm going to remind you of some things we've already covered, and when I'm putting this together as an entire lecture, right? So, paper published in Oncotarget, that's a journal. This is a research paper. See the reference there. Now, here we're starting to talk about specific proteins in the membrane. There's a type 1 transmembrane protein family member. So that means this family is large, or it has multiple components. And it's called the toll-like receptor 4. It's expressed, let me move this mug here, where this guy is, not only on immune cells, but also cervical and lung carcinomas. And also, I can tell you, on many other healthy cells. But when TLR4 is expressed in cervical or lung carcinomas, that's not where they're supposed to be expressed. So when you get a toll-like receptor expressed either on the plasma membrane or some internal membrane, because you know these toll-like receptors can work endomembranously or at the plasma membrane level, you're going to get a whole aspect of compositional changes and signal transduction cascades that are going to be completely nuanced by the expression of the toll-like receptor, essentially then pro forma generating the potential for an inflammatory response. Toll-like receptors, many of them, are linked with that. And we're going to get into all this as we move through the lecture, but you've heard it before. Okay? So that's why when you find a toll-like receptor 4 in a cervical or lung carcinoma at high levels, you know that that could well be a component of the oncogenic biochemical pathology. Right? So the expression of toll-like receptor 4 and the subsequent signaling cascade are indeed involved in making this other statement, okay, which has been backed up by the evidence of evidence in research science, involved in tumor growth, progression, and invasion. So all the way from the initial phases of tumorigenesis to metastasis. Toll-like receptor 4 signaling increased a particular enzyme, COX-2. What is that? Cyclooxygenase lysoform 2. And the product of cyclooxygenase is prostaglandin E2. Remember, that's an icosanoid, or sometimes a docosanoid. And by that, icosanoid means, of course, 20 carbon fatty acid that has been oxygenated. So these are oxy fatty acids. That's what icosanoids are. And one subclass of them are prostaglandins, and one particular molecular species is prostaglandin E2. Okay? So TLR4 signaling increases COX-2 expression and activity to synthesize PGE2, and PGE2 goes on to signal, and that may be linked to the early colorectal carcinogenesis, okay? While blocking at the same time apoptosis and further promoting angiogenesis. This is really bad when you're talking about carcinomas, right? Because you're blocking programmed cell death, which could save the system because you're killing the cells that are tumorigenic if you go through a rapid apoptosis and you don't induce inflammation if it's canonical apoptosis, unlike pyrotosis or ferroptosis or even necrotosis, right? But in this case, you block apoptosis so the cell doesn't die, plus you're promoting angiogenesis. So that means you're increasing the vascularization of the tumor. By that, I mean more blood flow. By that, I mean more oxygen. And yeah, respirable carbon sources like fatty acids and carbohydrates, thus feeding the growing tumor. Right? That's all bad. It appears to be all linked to toll-like receptor 4. Okay? So toll-like receptor 4 mediated cancer growth is also involved in breast tumor progression, and downregulation of TLR4 actually showed prevention of breast cancer progression and survival. Now here there's mostly animal models we're talking about. An animal is almost always rodent, right? Mouse or rat. But many of these studies have also been carried up into the clinic where there have been pharmaceuticals being tested to alter toll-like receptor for either expression or activity, sometimes even monoclonal antibodies to it, and then determining whether or not you can prevent breast cancer progression and survival. See, it's how the pharmaceutical companies learn from basic research science. And they go after those targets. It takes several years to come up with pharmaceuticals, bioorganic compounds, small active compounds usually, and whether or not they're going to function in the clinic is many years after that, right? Because there can be potential side effects. Further, toll-like receptor 4, and this is on membrane biochemistry. Remember, that's what we're doing here. I'm giving you detail as well as giving you a synthesis of all this information. I'm giving you the detail because this is what's necessary to understand it authentically. If you're taking a biochemistry course from me, I did this all the time. I dug into the primary research literature at the same time you may have I may have been giving the first lecture on glycolysis. Okay, sure, I'll tell you about hexokinase and glucokinase and phosphofructokinase and aldolase and glyceraldehyde 3-phosphate dehydrogenase and all the other enzymes, right? Pyruvate kinase. But at the same time, I'm going to give you that lecture. I'm going to go into some detail about how that particular pathway is regulated, say the Pasteur effect, ATP, citric acid, but also how it can be corrupted and talk about fructose, high fructose diet, for example, that you get from sucrose. Sucrose is a disaccharide, right, of fructose and glucose. When you take in fructose, even though fructose is a part of the glycolytic pathway, taking it externally is not the same as synthesizing it as a phosphorylated intermediate in metabolism. So I talk about what happens in high sucrose diets and the corruption of glycolysis, which is basically a what? Pathophysiology, right? Yep. Or I like to call it a pathobiochemistry because that's where it starts, right? Pathobiochemistry, and then the readout, right? The presentation of the disease is pathophysiology. See? Remember, we're biochemists here, and that's what we're up to. Okay, what else? TLR4 expressed in human lung cancer cells may play an important role in promoting immune escape by inducing an immunosuppressive cytokine flood and thus apoptosis resistance, while TLR4 functions in the pre-metastatic phase also of pulmonary metastasis. So this looks like a bad player. You might think, well, why doesn't the pharmaceutical company just come up with all kinds of great small molecules to interfere with TLR4, acting as partial agonists or complete antagonists, or even using just plain old monoclonal antibodies to just destroy that whole protein? Well, it's because TLR4 has normal biochemical use in the system. Very important biochemical. It's part of the defense system. So you can't just target something like toll-like receptor 4 with a pharmaceutical, because if you do, you're going to cause tremendous damage to the regulation of the entire system. This is why I don't like calling things side effects in pharmaceuticals. These are direct effects from pharmaceuticals. As precise as they can be synthesized or retailored after synthesis, small molecule chemistry going after specific protein targets like a toll-like receptor or something like a programmed cell death receptor on a lymphocyte. They seem to be something you would believe to be very specific and only knock out where the pathology is. Sometime that can occur if you can target it only when the pathogenic event is occurring, like in a tissue type, right? But if it's in your bloodstream because it was an injection and it's going through all the different cells and all the different subcompartments and organs in your body, including maybe the highly rarefied past the blood-brain barrier to the central nervous system, you can cause all kinds of tremendous damage by trying to knock out something like toll-like receptor 4 because it has all kinds of very important biochemical, healthy biochemical and physiological purpose. Okay. Again, I talk about this all the time, so I don't think I'm going too fast through it. Okay, so let's go through all this writing. I know it's a lot of writing, but you know, it's because I'm trying to be authentic with you. I'm not going to just say, I'm not going to summarize this in a sentence and then not give you the detail. Because what you can do with that, you can go watch, any number of other online lectures, and that's what they're going to do. I don't do that. I'm going to give you the detail. And the reason I write it out is, yeah, I know this detail, but I don't want to have anything be wrongly said. I want it to be precise and authentic. So I'm going to make sure it's in front of me. And I've written it and rewritten it enough times from the primary research literature that this is what the literature says, not something that I've got an opinion about. Right? This is direct dialectic. That's what we're trying to do here. Always stay logical, always move towards the truth when we're discussing biochemistry. So TLR4 expression is suppressed by epigenetic events. Okay, that's interesting, right? Now remember, epigenetic phenomena occur all the time. We used to think it was a rare phenomena. That means a covalent, usually modification of DNA, RNA, or protein like histones associated with chromatin that change gene expression. That's what we mean by epigenetic. So epigenetic is different than mutation. Mutation means you're changing the DNA sequence, right? You're altering the DNA sequence so you get different gene products or no gene product, right? Or at the transcriptional level, the same kind of thing, making transcripts that cannot be read out, translated, that is, to polypeptide because they're dysfunctional. Or maybe the polypeptide is made and it's quickly degraded, like an SOS response, an ER response, an unfolded protein response. You see? This is a different thing. Epigenetics means that real time, the environment, both external to the body and internal to the constant millions and billions of changes that are occurring when you consider all the cells communicating at the same time, from the moment of conception at embryogenesis to the moment of your death. That one living organism is constantly maintaining probably trillions of specific dialectical events to generate the ontology, which is just one living person, you. Okay? So I bring that up because it's so beautiful in the sense that it is extremely complex. And yet we know that all the complexity is necessary because minor changes in gene expression can completely destroy the health of the organism. And morbidity and mortality can be very rapid, right? A person can get sepsis even in the hospital and be dead in 12 hours. A healthy 25-year-old, right? Yeah, and that's not any external other than some kind of bacterial infection, right, that has gone too far. But living systems are very durable, and they're durable because of all this complex heterogeneity, which means complex control over all of these systems from every possible point of view, coming into and out of a specific enclosed reaction system. Right? Okay. So epigenetics is always happening. And basically, it's modifying gene expression in real time, that immediate response to stresses. And there's always stress, internal and external, in the body. And so those modifications of gene expression are basically how the system continues to function in real time and give you the longevity of 75, 80 years of life. And we didn't know much about epigenetics when I was in graduate school. We knew a little bit about some modifications of gene expression. We knew about promoters and enhancer elements, and even a little bit about splicing. Okay, this is like Stone Age, right? I mean, I got my PhD in 1983, right? That was really a long time ago. However, epigenetics was always kind of coming up because we were realizing that there were changes in gene expression, and yet the genes that were being synthesized weren't mutated. That is the product of the gene, the polypeptide. It's just that the change in gene expression could be induced by things like stress. So, and particularly in eukaryotes, particularly in complex multicellular heterotrophs like animals, particularly higher animals like mammals, and finally, the ultimate of all that complexity, human. Okay? All right, so epigenetic modification can occur at the methylation of DNA, which we talked about already many times. I'm going to give you a little micro summary of that in a moment, or at histone lysine residues, because lysine has the correct kind of polypeptide side chain that you can methylate. It's a lysine residue. You can methylate it or acetylate, or in fact, you can add other carbon-based small molecules that come right out of central casting metabolism to be covalently added to lysine, which modifies chromatin in real time to either, more typically than not, enhance global gene expression by creating the chromatin in a particular structure function relationship we call euchromatin. And euchromatin is more open to RNA polymerase, transcription factors, single-stranded binding proteins, and all the other repertoire that carries out transcription from DNA while unfolding it. Okay? Okay. So acetylation, for example, of lysine residues of histone usually means enhanced gene expression globally, not specifically. But it also can be specific depending on where the histones are located, wrapped around those histones with a chromatin, so that those particular genes that are proximal or distal to that region will be more prone to transcription. But you also can think about this as occurring for DNA repair and DNA recombination and DNA replication, the three R's, right? Recombination, repair, and replication, as well as transcription, can be enhanced by epigenetic phenomena. Think about that, right? I mean, that means the whole idea of how genes are expressed and how living systems work. It's not just DNA to RNA to protein. It was never that anyways, and we knew that in the 1960s from the elegant work done in Escherichia coli. But when we got into the animal systems, it was a whole new, not one order of magnitude, I would say 5 orders of magnitude more complex, easily. Okay? So, more detail. Epigenetic modification of CpG, that's cytosine, and it's a phosphodiester, to guanine, that's what CpG means. Those are 2 nucleotides, right? One is a pyrimidine, that's cytosine. The other is a purine, that's guanine, right? And remember, DNA and protein, and DNA and RNA, are made up of purines and pyrimidines, right? These are nucleotide bases. All right. Epigenetic modification of CpG islands has a major role in cancer. Okay. As methylation of that, wait for it, toll-like receptor for promoter, that means for the expression, transcription of the gene, is associated with toll-like receptor for silencing in a variety of cells, including intestinal, epithelial, and stem cell-derived vascular cells. So if you get silencing at the methylation level, you get a methyl group, a CH3 group, covalently bound to cytosine in CpG accumulated islands proximal to the promoter or sometimes enhancer regions or splice regions, junctions of a gene. Typically what happens, because that methyl group is there, it's a bulky hydrophobic group, right? It's a methyl group. You can't get the machinery in to do transcription. So we call it silencing. But guess what? The enzyme that added that methyl group, a DNA methyltransferase, we have DNA methyltransferases, which will remove the methyl group. And we also have demethylases. We have a whole family and class of enzymes which will add methyl groups and remove them. So think about how sophisticated that is. A gene has to be shut down because the system recognizes, because of all the trillion things happening all within 1 second, right? The whole living system put together. Enough signaling goes on to shut down the expression of a particular toll-like receptor in a particular cell type in a particular tissue for only maybe an hour, right? Or maybe for an entire cell cycle, and then put right back to what it was before the toll-like receptor is expressed. See how that's a lot more refined way to control gene expression than using like a monoclonal antibody to go in like a stone axe, right, and destroy the toll-like receptor. Because I said toll-like receptor is necessary in different tissue types, even when it's causing all kinds of havoc in a carcinoma, right, or in, say, I don't know, hypertrophy in the cardiac muscle, right, or atherosclerosis. There's all kinds of places where it can be. Expression of toll-like receptor can be pathogenic, but there's many, many, many other instances where it's not. Okay, this is just one example. Just one example. Think of all the proteins and all the lipids and all the membranes, right? Yes. Okay. So, silencing transcription via DNA methylation consists of two mechanisms. De novo methylation, that is, these CPG islands and promoter regions, they're not normally methylated for a given gene, but all of a sudden the signaling is right to methylate the cytosine residues on carbon 5 on cytosine. Okay? That's de novo methylation, or sometimes methylation mediated by transcription factor binding. So the latter will inhibit gene expression via repressor binding to the methylated CPG sites. How about that? So transcription factor comes into the nucleus already because of, let's say, neuroendohormone regulation bound to the receptor, like an estrogen receptor, moving into the nucleus and trying to turn all those estrogen receptor expressing genes, right? The ones that are responsible for expressing genes because of estrogen bind to its receptor and translocating the nucleus and working as a transcription factor. Sometimes through MAP kinase and kinase as well. All that's going down, everything's fine, but guess what? There's going to be a methylation that's going to block all that, and it's going to allow for what? A repressor, could be another polypeptide, to bind to that methylation site. And so the transcription factor can't do its work because it can't bind. So whatever genes were going to be turned on then and there cannot be. And the cell is constantly doing this to modify gene expression. And then again, you can remove all that hassle. You can remove the methyl group, and the repressor will just basically fall off because of thermodynamic considerations and maybe degradation following that. And so the gene then could be expressed just as normally, maybe 10 minutes later, maybe an hour later, maybe 2 cell cycles later, maybe next year, okay, depending on how long the cells last, you see? And all of those time signatures and all of that relevant biochemistry, we're just beginning to tease it out of nature, just beginning to. We have no model of the system. We just go in and find specific things like I'm telling you here and go, oh, wow, look at that. It's regulating toll-like receptor expression. But think about all the polypeptides and all the possible circumstances, and we just don't know all of those, right? All right. And of course, they can also go wrong. You can repress gene expression when you shouldn't repress gene expression because there could be a problem with the expression of the methyltransferase. Or maybe there's too much methyl donor group, acetylcysteine, in the nucleus, which can sometimes push the reaction towards methylation. Right? Now, here's where it gets down to something interesting. I mean, all that's interesting, right? Female hormone, 17-beta estradiol, that's the alcohol, promotes toll-like receptor 4 triggered. Here's what happens as a sequelae to TLR4 in most systems. Pro-inflammatory mediator production through a direct estrogen receptor alpha. Remember, you have estrogen receptors alpha and beta signaling in a specific cell type, macrophages. Remember that macrophages are antigen-presenting cells, and they can also, because they're macrophages, they can carry out phagocytosis and remove dead and damaged cells, or dead or damaged bacteria or virions, or parasitic structures, right? Yeast and fungi as well. Macrophages do all kinds of great things to clean up. But there are also the other function of macrophages is to carry out inflammation. Remember, those are part of the innate immune response. And they can also be antigen-presenting cells. So macrophages are amazing. They're in circulation, right? And they come from monocytes. And we talked a lot about that in the immunity lectures, right? Which I think now is almost 2 years old, but I went through 50 or 60 lectures on the immune response in humans. So just go back and listen to those, and then you'll know what I know. Okay, so methylation may diminish the protective role of inflammation. Here is where you want inflammation to work. And this is one of the ways that corticosteroids suppress the inflammatory response. Now, why do people take corticosteroids? Well, of course, if you have a surgery to, like, say, replace a liver, right? Or you get some other kind of organ replacement, or you're getting a lymphoma, complete readdition of all of your lymphatic systems, right? After destroying it and carrying out complete decimation of all the lymphatic structures and they're adding them back, you're going to want to immunosuppress, especially if it's not an autologous graft, right? Heterologous graft, maybe even from a relative, but not close enough to not be recognized as not self. So you suppress the immune response, right? Or like a heart transplant, it's another one, right? Okay. But people also take corticosteroids, what? When they have chronic inflammatory disease. Like what? Associated with what? Is that rare? Oh no. That's like people who are obese and have type 2 diabetes. They have chronic inflammation. And often people are put on corticosteroids. Here is what corticosteroids can do. See, they promote TLR4, they will promote TLR4 triggered pro-inflammatory mediator production through direct estrogen receptor signaling in macrophages. So the methylation will diminish the protective role of that inflammation. You see? So, following methylation, methyl CpG binding domains, proteins like MBD1, MBD2, MBD3, and methyl CpG binding protein, MECCP2, those are all different proteins involved in epigenetics, are recruited to the CpG site, repress transcription by recruiting another protein called SYN3A, which interacts with the histone deacetylase, the HDAC, to form a co-repressor complex. Okay. So here we're telling you that the female hormone can promote pro-inflammatory responses, but now I'm telling you how a methylation pattern, an epigenetic phenomena, can block that. Now, didn't I just tell you that corticosteroids are immunosuppressive? Yes, but not at every locus. Okay. Not every potential pro or anti-inflammatory response. You understand? It's never like that. You have contrarian and contradictory square of opposition events happening all the time, even around one particular neuroendohormone, female associated like this one, 17-beta estradiol, the alcohol, not the ketone. You can see that paper was published in Journal of Immunology, 12 years ago. Now, following methylation, we went through all that. So the transcription factor SP1 will bind to the TLR4 promoter, that's proximal to where you're going to start getting transcription, to potentially score target genes with prevention of CpG methylation, thus promoting transcription. So all that methylation, which made sure that SP1 couldn't bind, normally SP1 does bind to the promoter, which allows it to block the methylation, right? So there's a defense against methylation, but then there's also a counterattack of methylation by inserting that SYN3A, recruiting that SYN3A polypeptide. See how complex this is? So in complementary form, not farm, sorry, I misspelled there, methylation of cytosine in CpG islands within the SP1 binding site may directly suppress CP1 binding ability, thus shutting the entire system down epigenetically. Okay. So the differential DNA methylation of the TLR4 promoter in gastric cancer cell lines, which are expressing, remember I told you sometimes toll-like receptors expressed in tumors, expressing TLR4 at high and low levels, is an important mechanism underlying TLR4 transcription. With TLR4 silent cells showing increased methyl CP binding, and TLR4 upregulated cells showing enhanced SP1 binding to the TLR4 promoter. Okay. So you see how that works. The regulation of gene expression is constantly being rewritten with methylation pattern, thus to control either over or under expression of the toll-like receptor, this particular receptor, to mediate inflammation or block inflammation. So let's compare that and contrast that to the male hormone. Male hormone dihydrotestosterone, DHT, inhibits lipopolysaccharide-induced release of many pro-inflammatory cytokines. So there's a male hormone, not a female, inhibiting the bacterial cell wall lipopolysaccharide, which normally induces a pro-inflammatory response. It inhibits LPS-induced release of all these pro-inflammatory cytokines, tumor necrosis factor alpha, interleukin-1 beta, interleukin-6, also inducible nitric oxide synthase, and cyclooxygenase 2. We just saw that one. And then the product of nitric oxide synthase, nitric oxide, of course, right? And PGE2 come from COX2, like we just mentioned. In primary microglia, by suppressing the TLR4 mediated transcription factor, NF-kappa B, and then the MAP kinase P38 signaling pathway. Thus, ultimately, so you have to follow through the whole sentence, protecting neurons from inflammatory damage induced by activated microglia. Now, that's what the male hormone does in that system. Now, look when that was published. Neurobiological Disease, the name of the journal, right? In 2020, July, just 2 years ago, 2 years and 1 month ago. So we're still actively researching this. Did we know that beforehand? We had some idea, that's why the experiment was designed. But here it gives you all the detail. Now, of course, it doesn't mean that this particular research scientific statement will stand. We're going to continue to study this, and you better believe that we're going to find contrarian results and maybe even contradictory. And why is that? Because you're going to titrate that reaction with concentrations of receptors, all those intermediary signaling molecules at the level of transcriptional control, and even the hormone itself. Okay. But that's an interesting observation, right? So microglia induced in neuroinflammation plays a vital role in the etiology and the progression of neurodegenerative diseases, including Alzheimer's, Parkinson's, and multiple sclerosis. Okay, so that's why this research was done. So now we know something about what DHT can do. Now, if you use DHT as a prophylactic neuropharmaceutical, neuroendohormone pharmaceutical, does that mean, oh, well, now we have a cure for all these CNS diseases, these neurodegenerative diseases? No, because you better believe that the activity of DHT in the central nervous system is going to have pleiotropic phenomenological downstream sequelae. Okay. So this is where you need to study the whole system. Now, pharmaceutical companies often jump on something like this. In fact, it could have been funded by them. And they might say, okay, let's get some mimetics going. We'll try to get a DHT analog that could be easily degraded, or we can titrate easily in the bloodstream. And we'll just target it right into people that already have Parkinson's or Alzheimer's disease. But wait a minute. This research is being conducted looking at the microglia that's already activated. Maybe the prodromal stages of Alzheimer's or Parkinson's or MS will function completely differently in a different arena, because there'll be different macromolecular involvement and different membrane lipid raft biochemistry. See, so you have to look at all of the stages of the disease. And then you have to look at, could this work in women? Well, you need an androgen receptor. Women have deficiency in androgen receptors because they are not men. So if you don't have the right concentration of androgen receptors, how is DHT going to carry out this function in the CNS? Likewise with the estrogen. If you don't, estrogen receptors are far more populated in cells of a female than they are in male. So how will estrogen then be used in a male system that has really, really, really low levels of estrogen receptors? May not work at all. We may have all kinds of unwanted pleiotropic effects. Okay. I told you we were going to do some sex differentiation. We already got into it. Okay, so I'm going to just finish this page here, and then I think I'm going to stop because I'm sure we're moving right along here, and I got to keep my time because I've got an appointment coming up here pretty soon too. Believe it or not. All right, here's a paper published in 2020, Frontiers in Immunology. Toll-like receptors, of course, we know this, are innate immune receptors specialized in the detection of conserved molecular patterns, so they're pattern recognition receptors, present in pathogens. Now, the molecular pattern of pathogen is called a PAMP, right? Pathogen-associated molecular pattern. So it comes from the pathogen. And self-derived molecules released upon tissue damage, that would be from the host, and those would be called DAMPs, or damage-associated molecular patterns. DAMPs come from the host, for example, from the human. PAMPs come from the potential etiologic agent or pathogen. So, TLR family, which belongs to type 1 membrane glycoproteins, is comprised of 10 members in humans. Okay? TLR4 senses, as we just saw in the previous paper, lipopolysaccharide from bacterial cell wall, which is a major component of the outer membrane of the gram-negative bacteria, which exhibits potent immunostimulatory activity. Okay, so LPS will cause a tremendous immune inflammatory response when it's used to induce that. So TLR4 also recognizes DAMPs. Those are damage-associated molecular patterns. Those are caused by the host, right? Those are released upon tissue injury. Now, which ones? Well, here's one particular one I'm giving you an example as, i.e., high mobility group box 1 or HMGB1. We talked about this protein in previous lectures. Heat shock proteins, which HSPs, huge class of those. Reactive oxygen intermediates like hydroxide anion and superoxide. And extracellular matrix breakdown products is usually glycoprotein or peptide fragments. So TLRs, recognizing bacterial and fungal components, have been isolated according to their specific function to be TLR 1, 2, 4, 5, and 6. And they're expressed on the plasma membrane, while sensors of viral and nucleic acids, which are TLRs 3, 7, 8, 9, 10, 11, 12, 13, 12 and 13, are localized within the endosomal membranes. And TLR4 is actually mobile. It can move from the plasma membrane to all those endomembranous compartments, as I told you a half an hour ago. So the association of TLRs with their specific ligands initiates intracellular signaling routes through the adapter MID88. Okay? Sorry, that's my outdoor clock. It's a bird noise because it's 5 o'clock here. So the association of TLRs with their specific ligands initiates intracellular signaling routes. Okay? Through the adapter, here's an adapter peptide called MID88, except for TLR3 that signals via another protein called TRIF. Okay? This culminates in the induction of pro-inflammatory cytokines via the transcription factor NF-kappa B, or via activation as an antiviral interferon. So interferon is another way to deal with intracellular pathogens, and it's more specific to virus because it has a process where viral replication can be blocked. That's why it's called interferon. Interferon was first isolated as a nucleic acid mechanism to interfere with the replication of viral genome intracellularly. Okay? So aberrant TLR4 activation, move myself out of the way, has been associated with chronic inflammation disease, as you might guess, as we've been saying, because it's pro-inflammatory. So a significant amount of evidence suggests that TLRs are associated with specific diseases. That is sepsis, asthma, AIDs, autoimmune diseases, cancers, diabetes, intestinal disorders, cardiovascular diseases, and finally neurodegenerative diseases. So that you can believe there's a great deal of interest in understanding how toll-like receptors mediate pathobiochemical events. And that's why I spent some time talking about them on just the membrane lectures, the membrane biochemistry lectures. That's why I recapped and synthesized that information here this afternoon, this beautiful Sunday afternoon. Okay? So I'm going to stop here because it's been about enough time. I told you I had a prior engagement. And I want to just basically end, flies outside, by saying that this 44th lecture in membrane biochemistry was what I call a synthetic lecture. It took all the analysis that we've been going through in the lectures before, although I mixed my synthesis with my analysis. Sorry, I'm not any better a metaphysician than Immanuel Kant, did the same thing, so there. But this is more synthetic than it is analytic, meaning I'm trying to put together aggregate concepts and ideas, as well as giving you specific details of biochemical events. Okay, so just scratch the surface with this lecture. This is going to be at least 2 more videos, so probably an hour each. Video lectures go a lot slower than audio because I'm showing you pictures and I'm talking to you and I'm thinking about more things as I'm talking. The audio lectures, I really try to do more very, very drilled down. Let's get this 30 minutes with as much information as possible. Sometimes I don't succeed in that either, but anyway, basically that's why we did this lecture today, is to give you a chance to see video. And if you want more video lectures, let me know, because I don't really get any feedback except people online will say, hey, I really dig your video lectures. Okay, well, that doesn't tell me much detail. When you see this lecture, go ahead and write something very specific. Now, I have had people ask me very good questions, and I've tried to respond to them. But what I'd really like to do is have many more video lectures and have it so that we can do some real online teaching at the graduate medical school, pharmaceutical school, veterinary school levels of about subjects associated with biochemistry. And in order to do that, we have to really build up the series of lectures, which means we have to have, you know what I'm going to say, some kind of contribution. Because it takes a lot of time to build these, and I'm a person that worked completely on my own. I write all these lectures, read all the literature, and then of course I'm producing them and I'm hosting them. I'm delivering them. I like doing that, but it takes away a lot of time that I would be doing something else. So if we can do more video lectures and more audio lectures, please subscribe to my YouTube channel where you'll see this. Please subscribe to my podcast channel, and please contribute. Okay, there's plenty of ways to do that. So I will stop here on this beautiful 28th of August, 2022, on Sunday afternoon in the Inland Pacific Northwest, which you can kind of see behind me there, the pine trees. And I guess I'm going to have to figure out how to get this done here. Yeah, I know how to do it. I was kidding. So goodbye for now.